Okay, welcome back. This is going to be our second video of our three-part review. First part was on properties of water. Second part, this part is going to be on macromolecules. And the third part, I'm going to be walking you through an FRQ that you may or may not see a similar FRQ on your exam this coming week. So this PowerPoint was introduced by asking us what is the other 40% of life if it's not the 60 to 70% that is water. Well, and hopefully you remembered learning about macromolecules. Now, I provide this slide again because if you have filled out that chart in a great amount of detail that I gave you at the beginning of the unit where it asks you about functions, cellular locations, how you identify it, then you're already in a pretty good shape for the exam. I'll be really frank that the exam is really going to be asking you how can you tell one apart from another based on some sort of information they give you. So if you can know all of the differences between all the different macromolecules, you'll be in pretty good shape as long as you know how to apply it in maybe a scientific or a laboratory setting. So start thinking about, okay, if I see sulfur in a sample, I know that I'm probably dealing with proteins. If I see phosphate, I know that I'm probably dealing with nucleic acids. If I have a carbohydrate, or I see a 1 to 2 to 1 ratio, I know that I'm dealing with a carbohydrate rather than a lipid. Those are the sort of things that AP is really concerned with. And so as you are studying, it's not just a matter of memorizing things, think about how you could actually use it in a lab. Okay, so we talked a lot about macromolecules, but the reason why these macromolecules are going to be carbon-based is because carbon always makes 4 bonds, and that's because of how many electrons it has in its valence shell. Now, I'm never going to ask you that, but it is very important that you understand that the carbon is almost always going to make 4 bonds, especially in our context. If you go take organic chemistry, you'll start to learn some differences, but for our purposes, just know that carbon is going to make 4 bonds. So if you see a carbon not making 4 bonds, it either is going to be secretly bonded to a hydrogen, meaning that they just haven't drawn in that hydrogen, or it has to be charged, which we have not seen that, and you probably won't see that until you go into organic chemistry. So for our purposes, it's always making 4 bonds. If you don't see all 4 bonds, then it's probably to a hydrogen. Okay. So because carbon is able to make 4 bonds, there's not a lot of elements that want to make 4 stable bonds like that. For example, oxygen only wants to make 2, nitrogen only wants to make 3. So this makes a really good scaffold for all organic molecules or all life molecules, right? And so we looked here first at these organic molecules, and we looked up butane and isobutane, and I remember asking you if we thought that they would have the same function. And they do have similar properties. They're both going to be nonpolar because they only have carbons and hydrogens, but they're not going to behave the exact same because structure matters. So it's not just the composition, meaning the atoms in there, it's also going to be the structure. And so that's when we started talking about isomers. Isomers can have lots of different types, which you will talk about in organic chemistry if you go and take it. But for our purposes, we focused on structural isomers, which are going to be those isomers that have the same elements in the same amount, but different connectedness. So we looked here at 2 different compounds. One is valine, which is not really a drug, it is an amino acid, and it's very good for us. We need it to build muscle, we need it for lots of different biological functions, and we compared that to the drug amyl nitrate, which is just a party popper. If you take too much, you can die. So, same compounds or same elements, but different connectedness can have a very, very different effect on our bodies. Okay, then I wanted to remind you of this idea of functional groups. Throughout this course, I'm going to be mentioning these functional groups by name. If you don't have these memorized, you can get really lost because you're more stuck on what exactly is an amino, what exactly is a carboxyl. If you have these committed to memory, you're able to follow along in the conversation a lot more easily. So that's really why we want you to understand these functional groups, and we also want you to understand that because they're always going to have the same connectedness, right? A carbonyl is always going to have a double bonded C and O. Carboxyl is also going to always have that double bonded C and O, but also that additional hydroxyl group. And so they are going to have similar functions from molecule to molecule because it's the same connections, same elements. Okay, and then we started moving into directly macromolecules, macro being big and molecules obviously meaning multiple atoms. And a lot of this is going to be reviewed from when you took pre-AP Bio or when you took Bio Honors. If you had Bio Honors, if you were in my class, you would have, most of this would have been review. If you did not, then probably not. So some of this would have been new, but a lot of it could still be review. So keep in mind what you've already learned and then make it a little easier on yourself by recognizing the things that are new. All right, but to make a macromolecule, well, technically the monomer is considered a macromolecule, but usually they're going to form polymers that are really interesting. And so how do we form a polymer from a monomer? Well, we are going to have to use dehydration synthesis. So I remind you again that this is going to be a very important process for forming bonds between all of the macromolecules. So in all of the examples that we saw at least one situation where dehydration synthesis was used to form a bond. And then we talked about how you can reverse that, which was going to be hydrolysis. If we add water back in, then we can reform the hydroxyl and a hydrogen on the other side. So remember, hydro means water, lysis means to break. And again, this is going to be facilitated by enzymes, because if every time that my proteins or my carbohydrates were subjected to water, if they were just able to break apart, then I would never be able to get highly ordered life. So we really need enzymes to be carrying out this process and regulating it. Otherwise, I would be always breaking down. So very, very important that dehydration synthesis or condensation, same thing, is going to form bonds. Hydrolysis is going to break bonds, and it's involved in all of the macromolecules. I'm going to skip this slide because it wasn't really necessary for this exam, or the idea is just that it's very important to understand that we have really, really diverse structures because we can make lots and lots and lots of huge molecules by putting together lots and lots of different monomers. Okay, so then we moved into carbohydrates specifically. So carbohydrates are famous for being short-term energy, but maybe you didn't think about before that they're also really important building materials. So this is especially true for plants. So plant cell walls are going to be made up primarily of carbohydrates. They also can be used for plants to grow. So very, very important for both of those. Yes, important for short-term energy, but don't forget that they are also building materials. We call the monomer a monosaccharide, and then when we make lots and lots of monomers, we put them together, then we have what we call a polysaccharide, or aka a polymer of a monosaccharide. So these monosaccharides are almost always going to be in a 1 to 2 to 1 ratio, and that means that if I had 5 carbons, I'm going to have 10 hydrogens and 5 oxygens. That keeps in that 1 to 2 to 1 ratio. And so that means that equal number of carbon and oxygens, double the number of hydrogens. So you really only need one of those. If they tell you it's a carb, you can predict the other two if they tell you it's a carb. Now we have carbonyls and hydroxyl groups involved in the monosaccharide. So very important. Also notice that these are linear structures of monosaccharides. Don't always just rely on looking at a ring. I also told you a big key trick is if you see a word that ends in "-ose, it often is going to be a carbohydrate. So we have, not all carbohydrates are going to end in "-ose. We have glyceraldehyde shown here. Dehydroxyacetone is also a carbohydrate, and we're going to talk about one more, pyruvate, when we get into cellular energetics or unit 3. So it doesn't guarantee, if it doesn't end in "-ose, it doesn't mean it's not a carb, but if it ends in "-ose, it probably is. Okay, so we usually name molecules based on their carbon structure. I'm never going to ask you to name a molecule, and neither will the AP exam. That is not something that would be for this course, but if you end up being very interested and you do want to go into organic chemistry, you'll spend a lot of time doing that. Now we talked about how you might see a linear structure, and if you see a linear structure, that means that it could be a carb as long as it follows that 1 to 2 to 1 ratio. So you'll need to count and see if it follows a 1 to 2 to 1 ratio. But also note that in aqueous solutions, they often form rings. So know that, recognize that, but also don't use that as your telltale sign that it is going to be a carbohydrate, because we're going to see ring structures with lipids as well when we talk about steroids. Okay, now really only for carbohydrates and really just carbohydrates, we talk about disaccharides or the dimer, monomer, dimer, polymer. The reason why, I guess, is because there are two very important disaccharides that are kind of interesting, right? Sucrose is the one that we eat, that's table sugar. Maltose is the disaccharide of glucose. And then lactose is also important because it joins glucose and galactose. When we talk about the lac operon, that'll be a little bit more important, but that is not going to be for a long time. So don't worry too much about this. You do not need to know these structures. Just know that there's going to be a glycosidic linkage between two monosaccharides to form a disaccharide via dehydration synthesis. Okay, this was the fun stuff for me. I think this is really neat. So we talked about some polysaccharides, and we focused on polysaccharides of glucose. So we talked about starch and glycogen. Remember that starch is going to be in plants. It's a plant storage polysaccharide. And glycogen is going to be an animal storage polysaccharide. It is stored in our liver and our muscles. Now the difference between them, because they are both using glucose and we're able to digest both of these, the difference is the structures. So with starch, it's going to be a linear polymer of glucose, and glycogen is going to be a branched polymer of glucose, which makes glycogen easier to break down because our enzymes are able to access a lot more ends. And I hope you can remember the picture that I drew out on the board, so that makes it a little bit more helpful. But remember that this is really helpful for animals that have to have very, very quick senses and be able to burn energy very, very, very quickly, whereas plants don't really have to do that because they don't move. Then we talked about a third polysaccharide of glucose, but this one is slightly different. This is cellulose. This is going to be a structural polysaccharide, not a storage. It's never really getting broken down for energy, and we can't break it down for energy either. So if it is a polymer of glucose, why can't we digest it? Well, it turns out the glycosidic linkage between the polymers are different. So that's a lot of words to say that they use beta glucose for cellulose, and they use alpha glucose for starch. When I say they, I mean plants. And so cellulose is what we call dietary fiber. It's still really good for us. We should eat it, but it is not going to be digested. We are not going to be able to use it for energy. Then the last polysaccharide that we talked about is chitin. This is actually not a polymer of glucose, but it is a structural polysaccharide. It's going to be found in exoskeletons of arthropods, so something like an ant, grasshoppers, anything like that. And it's also the structural support for cell walls and fungi. Okay, then we started talking about lipids. So lipids are kind of, in my opinion, the most boring in the sense that there's not that much to talk about, but they do taste really good, so that's nice. What makes lipids a little different is that they are going to be insoluble in water because they are, remember, mostly hydrocarbons. If they're mostly hydrocarbons, that means that the bonds between hydrogen and carbon are mostly nonpolar bonds, which means that polar substances like water are not going to be able to dissolve it. We say that they don't really have a true polymer. If you're looking for one, you might call a triglyceride a polymer. If you're looking for a monomer, you might call a fatty acid a monomer, but you really don't hear that terminology very often, and it's not going to be tested. Now, there are three families of these lipids. We call them fats, phospholipids, and steroids. Fats are going to be a glycerol and a fatty acid, and we form the bond between the fatty acid and the glycerol using, once again, dehydration synthesis to generate an ester linkage. That's the name. It's an ester linkage. Now, these fats are mostly a leftover mechanism from when we weren't really very good at hunting. So when we did get a nice big meal, it would be advantageous to be able to store that energy. And so we did. Our body stored it as fat. And then if we were in a starvation state, then we could burn that fat so that we didn't immediately perish. So it's very important for extra storage. It also cushions our organs and insulates our body because Fat tissue is not metabolically active, which means that it is basically not taking any energy from us, so we can just kind of use it like a blanket. Okay, this is a discussion that I really want you to know. This is a wink, wink, nudge, nudge to your exam, but it's very important to me that you know this, that the difference between saturated and unsaturated fat is really down to these two bonds, or the double bonds. So in unsaturated fat, we have what we generate a kink, meaning that the structure physically bends, and that makes it so it's very hard to stack these structures. If we look over here at the unsaturated triglyceride, if you were trying to stack a bunch of these on top of each other, let me use my PowerPoint pointer, this thing here, if you were trying to stack a bunch of these on top of each other, you would have a really hard time. And so that makes it liquid at room temperature because it's literally not as dense. Now, saturated fats, they don't have any double bonds, and so they lay pretty flat. They basically look like a rectangle. And so these stack super, super easily, and then therefore they are able to be solid at room temperature because it's much more dense. Typically, we say that saturated fats are going to be less healthy than unsaturated fats. When we get into metabolism, we might see why, but that's not really what I'm worried about. What I do want you to start thinking about is, remember that our membrane is a lot of fatty acids because it's going to be a phospholipid. It's that phospholipid bilayer. Now, we don't do this, but bacteria have a mechanism that allows them to convert their fatty acids from saturated fatty acids to unsaturated fatty acids, and vice versa. They have really specialized enzymes that are really good at this. And so what I want you to think about is how that could be used as a form of temperature regulation. Think about if it's really hot outside, which one would they prioritize? Which type of fatty acid would be better for their membrane? Or if it's really, really cold outside or their environment, which one would be more advantageous, saturated or unsaturated? Start thinking about that. I'm giving you a little wink, wink, nudge, nudge, but very, very important concept, and I will be asking you something about it. Now, my favorite lipid is going to be the phospholipid because it's going to be very, very important to our cell membranes. Now, recall that a phosphate is going to be one of our functional groups. And hopefully, by now, you know that phosphates are charged, and we can see that because we have a full negative charge here. We're not going to call this polar because polar is about partial charges, and charged functional groups are going to be full charges. So this is a full negative charge. Now, I told you that you're not going to see this structure probably ever again in biology because we're a little bit lazy, and instead we draw it more like these little ghostly structures with a little phosphate head in purple and then the fatty acid tails, and I guess that kind of reddish brown color. Now, this term amphipathic means that there are going to be both polar and nonpolar portions to the molecule, and these are going to be very important to our cell membranes. Inside the cell is a lot of water. Outside the cell is also a lot of water. So we actually have to have this bilayer, otherwise the fatty acids would be exposed to water, which is what they really, really don't want. Remember, they are nonpolar. They do not like water. They are hydrophobic. So they are going to have to form with other phospholipids and orient themselves so that only the head group is going to be facing water and the fatty acids can exclude that water. Very, very important concept. Then the last group that we talked about was steroids, and this is the one that I'm going to tell you, if we see rings, we can't assume it's a carb because we see rings here too. Typically it's going to be 4 rings connected together, and that is different than carb because the carbs are not going to be sharing bonds like this. But it's kind of hard to notice that when you are early in your biology career. So I just kind of don't like to even worry about ring structures as an identifying feature. Instead, I like to worry more about that 1 to 2 to 1 ratio. Now I want to bring this one up again, that remember that every time you see a point, that is going to be a carbon. Carbon always makes 4 bonds. So this carbon here, where my pointer is pointing, has 1 bond here and another bond here. But if it makes 2 more bonds, well, those are just going to be assumed. Organic chemists are a little bit lazy, and so they assume that you know that the other 2 bonds, they'd be off where I'm kind of pointing back and forth, they'd be off with hydrogens. So they would be always saturated with hydrogens if we don't see other elements that it is bonded to. So keep that in mind. These are not carbons making 2 bonds. They are making 4. We just don't draw in the carbon or the hydrogens. Okay, then we started talking about proteins, which are my favorite macromolecules. They are just going to be so special. We're going to talk about so many this whole year. They are so cool, and they are so diverse because they're huge. They are absolutely ginormous, and they perform almost every function except genetic material. So they are important for structure, storage, movement, defense. Our antibodies are proteins. They regulate our metabolism. They regulate transport, communication, and there are receptor proteins. So very, very, very important. You can't get away from these. These are going to be underscoring basically all of biology. So very, very, very important. Okay. So I hope when I introduced functional groups, I impressed upon you that it's not an accident that we named amino acids the way that we did. They always include an amino group and always a carboxylic acid group, and that is going to be the acid portion of amino acid. So this top part of the structure, anything above the R, is staying the same for every single amino acid. All of them. We have 20 different amino acids, and all of them all have this top portion. What is this different is going to be the R group. This is going to tell us the identity of the amino acid, and we'll talk about some of those identities in a second. But very important that you know that this top part is going to be the same. Therefore, we can look for this NCC structure. NCC, 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 NCC repeats, and we will be able to tell that that is definitely a protein. The linkage between these adjacent amino acids is called a peptide bond, and believe it or not, we form that peptide bond once again through dehydration synthesis. Now moving on to these R groups. So these R groups are highlighted in yellow, and I do not want you to spend any time memorizing these structures. It would take you a very, very long time to get it perfect, so it's not worth it. You might do it in biochemistry if you take that, but you will not have to do that ever in this course. But what I do want you to know is that these R groups are going to determine the type of amino acid it is. So is it a nonpolar amino acid? Is it a polar amino acid? Is it an aromatic amino acid? Positively charged, negatively charged? That is dependent on the R groups. And I also want you to start thinking about, okay, if I mutate my protein, going back all the way to the DNA level, I change the codon so that it codes for a different amino acid, you should start thinking, okay, is it going to have a big effect? Well, if I go from a polar to another polar, something like asparagine to glutamine, well, those are roughly the same size and roughly equivalent in terms of polarity, so it's probably not going to have a huge effect on my protein. But if I convert it from a polar, maybe a serine, to a nonpolar valine, well, now we're going to have a situation where that could have an effect. That could affect the folding, which we're going to talk about in a second. It could, if that is an important amino acid for some sort of mechanism, then we could have an issue that that mechanism doesn't work, and that could make the entire protein not work anymore. So when we go from polar to nonpolar, then it could have a very serious effect. Same thing if we go from positively charged to nonpolar or aromatic. Changing the group can often have a very large effect. Now, also staying within the group could have a big effect. Just because it's polar doesn't mean that we get to do anything we want. So It could still have a very large effect, but it is less likely. It is less likely to have a big effect. So keep that in mind as we are moving through and you're preparing for your exam. Always think about what the applications and the implications are. Okay, so I told you again that this is called a peptide bond that forms when we take out water, dehydrate, and we synthesize a polypeptide. So we call the bond between the two amino acids a peptide bond. So hopefully you notice here that we see that NCC, NCC, NCC repeat, and that will go on till the end of the protein. Okay, now what makes proteins so versatile is the fact that they form a 3D conformation. So this is very important. Shape determines function, or you'll also hear me say all the time, and it's a theme in biology that I never want you to forget, that structure dictates function. That if I know the structure of something, I might be good at picking or predicting what that protein does, or what that carbohydrate does, or what that lipid does. That structure dictates function. Now, the other macromolecules that we have talked about really don't form this extreme of a 3D shape. I'm not going to say that they're not 3D. Of course they are. They're still atoms and they're still living in our world. But when I'm talking about proteins, I'm talking about full folds that are massively, it's a massive linear structure that can fold on top of itself and really make really interesting and different shapes. We're going to talk about lots of very important enzymes and how their shapes end up affecting what they do. I'm very excited to start talking about ATP synthase, but for right now, just know that structure dictates function. And also know that your DNA, remember DNA is ultimately going to code for proteins. Not directly, because DNA has to code for RNA first, and then RNA will code for the proteins, but it is ultimately coming from the DNA first. So the first structure that we have with proteins is the primary structure. This is the sequence of amino acids. So it is not getting into a folded structure at this state. If you sequence, which many of you might do if you end up going into science for your career, you'll sequence stuff all the time. And what you'll get back is going to be a bunch of amino acids. They say it's a lysine, it's a valine, phenylalanine, glycine. And it'll tell you the order of those amino acids. Now, that is not telling you anything about the shape of that protein. That is just telling you what amino acids and how they are linked together. This is, again, of course, determined by the DNA. Then it's transcribed into RNA, and then you read that codon chart, and you get the amino acids. So hopefully you notice here that the structure is always going to be NCC, 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 and the identity of the amino acids is told to us by that R group, that group coming off of the middle carbon. The secondary structure is not true folding from one end of the protein to the other. Instead, it's kind of like a small fold that is occurring really locally. This is going to occur because of regular hydrogen bonding along the polypeptide backbone. So hydrogen bonding is going to naturally just going to occur, and that is going to result in two types of secondary structures, the alpha helix and the beta pleated sheets. So the alpha helix is the one here. It looks like a helix, so it's named very well. And then the beta pleated sheets are right here. They also kind of look like sheets. They kind of look like if you do an accordion of your paper, then you have a beta pleated sheet. Okay, then we move into the tertiary structure, which is going to be where a lot of proteins stop. So not all proteins go into that fourth one, which we're going to talk about in a second, but many proteins stop at the tertiary structure. And this tertiary structure is now when we're getting these big folds. These are going to be big, big folds, potentially from across the entire protein. Now, not always. It could be localized folds as well, but they're not going to occur from regular intervals of hydrogen bonding. That would be your secondary structure. Instead, these are going to be big folds that are formed typically through hydrogen bonds. So we can see that here from other amino acids. So you can imagine this looks like a serine to me, and then this looks like maybe the carboxy terminus. And so that can generate a hydrogen bond. You don't need to know that that's a serine, but know that we are capable of doing hydrogen bonds that allow for large folds across the protein. We also will have disulfide bridges. Remember that there are two amino acids that have sulfur in them, cysteine and methionine. Well, cysteine can find another cysteine somewhere else in the protein and say, hey, I'll give up my hydrogen if you do too, or really give up my proton if you do too. They leave behind their electrons, and then now we have two electrons, and that is going to form a true bond. We have a shared pair of electron here. We call this a disulfide bridge. Make sure you know this term, and make sure that you know that it's important for tertiary structures rather than secondary. I've seen students make mistakes on that before. So disulfide bridge, di meaning two, sulfide meaning sulfur. It's not an accident that it's named that way, and bridge being the bond between these two sulfurs. We also can have ionic interactions because remember we have charged R groups, those positively charged and negatively charged R groups, and so we can form an ionic bond. These guys really like to hang out together because this is a full positive and this is a full negative, and this is very important. And then also hydrophobic interactions. So we have some of those non-polar R groups, which they don't like water, so they will find each other in the protein and they'll say, hey, let's hang out and let's get rid of water. So they'll kick out water and they'll have to kind of be next to each other in order to successfully do that. So all of these different ways we can get a very folded structure that is ultimately going to determine function. Now the last one, not all proteins do this, but they can. Not all proteins can, but some of them can. And this is where we're going to have multiple polypeptide chains coming together to form basically a super protein. So the example I gave you in class was going to be about this myoglobin and hemoglobin protein. So myoglobin is its own thing. It can be coded for on its own. And it's actually pretty good at carrying oxygen, but not nearly as good as hemoglobin. So when these four myoglobins find each other, they can form basically a super protein called hemoglobin and carry oxygen throughout our blood. So really important that we understand that this can happen and that some proteins do this. Many proteins do this, but they don't have to. Okay. This idea, structure dictates functions, comes everywhere in biology. We are going to see it all throughout the entire course, so please don't forget it, that structure dictates function, that the function of the protein is coming from the architecture of the molecule. Very important that you know this. So with that, if it's only really its true protein once it's folded, if we unfold it, it's no longer going to be a functional protein. So we call this process denaturation. And we talked a little bit about denaturation with DNA as well. That is really more for unwinding the DNA, but that does not mean that it can't go back. We're going to talk about that when we get there. So just keep that in the back of your head, but don't stress out just yet. But denaturation is where if you subject a protein to conditions... Sorry, the bell. I'm doing this during my planning. If you subject the protein to conditions that it doesn't like, something like a higher temperature, changes in salt concentration, changes in pH, then it will unfold. It doesn't like that. So it'll unfold, and it's very hard to renature. Can it be done in a cell? Yes. In the lab, it's very, very, very hard. So we typically just throw out that protein. But yeah, it's typically not a good day when you accidentally denature your protein. And a lot of times, enzymes really should be kept frozen because they won't denature that way. If you keep them on your bench top, it might be okay, but a lot of times it will end up with a denatured enzyme, and then you'll wonder why did my experiment not work? Well, I wasn't using a functional enzyme. Okay. So on your own, I want you to just try to go through and see where you're getting stuck. If you can't really just sort of repeat what I just told to you, at least, and understand everything that I said, you probably need to go back and study a little bit more and make sure that all of that is clicking before you move into really the next unit. The next unit, we're moving fast, right? Okay, so I talked about enzymes. Enzymes are my favorite types of proteins. They are the master regulator in our bodies, and they are very important because they make things biologically feasible. They can't change the thermodynamics of a reaction, so the reactants and the products really have to kind of look like this for the reaction to go without the input of energy. But what they can do is they can lower the activation energy, and that makes everything go by a lot faster. So these are what we call biological catalysts. You might notice that they often end in ACE, and so that's a big hint that you're dealing with an enzyme. And I also wanted to start thinking about, do you think because we don't use up enzymes, do we typically need more enzyme or less enzyme than the substrate? If I had a lot of substrate, could I successfully digest all that substrate with just a little bit of enzyme? Start thinking about that as you prepare for your exam, although it's not going to be a huge portion of this exam, but when we move throughout the course, start thinking like that. Okay, so we ended our discussion with nucleic acids, and this is probably an area where it's mostly review, but I do need you to know a couple of new things, which I'll bring up again in a couple of minutes. But know that there are two types, DNA and RNA. The difference is going to be multiple differences, which we'll talk about in a second. DNA is a double helix, RNA is a single helix. They follow, or DNA follows, the Watson and Crick base pairing system. You can have double-stranded RNA, but we're not going to get into that because that's really beyond the scope of our course. But know that it's not impossible. Some viral genomes are going to be double-stranded RNA. Oops. We call the monomer of a nucleic acid, which nucleic acid is our polymer, DNA or RNA. We call the monomer a nucleotide. Very important that we know that. Now, we also have some vocabulary that is important in this unit, and it's going to be very important as we move in through the course, and that is that chromosomes are going to be made up of DNA and actually protein as well. I told this to one of my classes, and I thought it was kind of interesting, so I'll let you all know now that DNA, remember, it has a phosphate group to it, and since DNA has a phosphate group, phosphates are negatively charged, so DNA's overall charge is negative. And so that doesn't seem like that should work, right? It doesn't want to then fold up and be condensed, which it has to be. If it wasn't folded up, we wouldn't be able to fit it in our bodies. So it has to be folded up. So how does it fold up? Well, it has to fold up around a bunch of proteins. Well, if DNA is negatively charged, these proteins that it folds up around are actually positively charged because it can sort of balance that negative charge, which I think is pretty cool. You're not going to be tested on it, but it is something maybe we're thinking about as we move through the course about how things can possibly work if we know some chemical properties about it. Okay, so there are three parts to a nucleotide. I mentioned one in that last slide, just sort of secretly, that there is a phosphate group. Well, there's also a sugar, right? Deoxyribose, we see that suffix "-ose" again, so it's going to be a sugar. And ribose is in RNA. Now, just like if you dehydrate something, you take out water. If you deoxify something, you take out oxygen. So we have one fewer oxygen in the DNA sugar, deoxyribose, than in ribose, the RNA sugar. So that's a huge difference between DNA and RNA. And then also we have a difference in bases, thymine versus uracil. Okay, so now we know that the three parts of a nucleotide are going to be the phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. Remember, I told you to repeat it over and over and over again. Now, the sugar can be different, right? And it is. In DNA, it's deoxyribose. In RNA, it's ribose. The phosphate group stays the same. That's the same in both of the molecules. But the nitrogenous base or nitrogen-containing base is different. A, G, and C is in both of them, but T is only in DNA and U is only in RNA. So if I have a question on the exam that asks you how much T is in RNA, don't just immediately try to subtract from the other percentages given to you. Think that question through. Think, okay, RNA doesn't have T in it, and so I can immediately pick 0%, and vice versa. If I give you a DNA strand and I ask you how much uracil, the answer is just 0%. Now we also have some new vocabulary words, purines and pyrimidines. My way of remembering it is pyrimidines cut. So C, U, and T are my pyrimidines, and my purines are A and G. Now, at the end of the day, I'm not really going to ask you for these structures, but it might be helpful to remember that A and G are purines and C, U, and T are pyrimidines. The reason why is because sometimes we start classifying DNA strands and RNA strands as purine-rich or pyrimidine-rich, and it can kind of become important when we start thinking about like molecular weights. But for right now, as long as you know which ones are which, that's totally fine by me. Okay, so we come back to this idea about dehydration synthesis, that we form bonds by taking out water when we're talking about these macromolecules. And we do. So between the N base and the sugar, and between the, N base and the sugar, and between the sugar and the phosphate, those are all covalent bonds formed through dehydration synthesis. But if we have, remember, DNA is double-stranded, and so we have to be able to form bonds between the strands. Well, these bonds aren't true bonds. Instead, they are going to be hydrogen bonds, which are significantly weaker than covalent bonds. Well, if they're significantly weaker, that means that it's actually a whole lot easier for me to, what we call, unzip the DNA during times where we have to unzip it. For example, replication, transcription, all of these terms that you've learned before, now we're putting it into a chemistry context, which I think is really cool, right? But one more piece to this is that we need to start thinking about how many hydrogen bonds. Well, if the strand is GC rich, meaning we have a lot more GC than we do ATs, well, GC forms 3 hydrogen bonds, AT forms 2 hydrogen bonds. So if I have a GC rich strand, that means I am going to take more energy unzipping this DNA. Now, our cells do it. Our cells will have to unzip DNA all the time for replication. But we also sometimes like to do this in the lab. If I want to amplify my DNA or replicate a DNA sample, I can do that, but I do need to know the GC content. Because if my GC content is really high, I might need to raise the temperature during that step, during PCR. And we talked a little bit about that in class. I don't expect you to know the steps of PCR just yet. We will talk about them later. But I do want you to start thinking about, okay, if there is a step in PCR where I have to unzip the DNA and I can change something about that step, aka change the temperature, think about if it's GC rich, if I need to raise it, or if it can be lower. That's very important. Remember, AP Bio, all about application, all about implications. Okay, hopefully you all know the base pairing rules at this point. When I asked you in class, it seemed like everybody did. A pairs with T, C pairs with G, and in RNA, A pairs with U. And remember that these are connected with hydrogen bonds, and it allows DNA to make exact copies of itself. If we had covalent bonds, it would take way more energy to separate the 2 strands to use one as a template. So we really have to have these weaker bonds or hydrogen bonds to be successful in that replication. All right. Now, I do expect you to know the differences between DNA and RNA. There are 3 major differences. The sugars, right? Deoxyribose versus ribose. There is 1 fewer oxygen but in the deoxyribose sugar. And there is a difference in the strands. DNA is double-stranded, RNA is single-stranded. And then the bases. So the bases are A, G, C, and T in DNA, and A, G, C, and U in RNA. So those are the chemical differences. The functional differences is that DNA actually does encode directly for protein. It codes for RNA. And it can code for a lot of different types of RNA, which we'll talk about when we get there. And RNA codes for protein. This is really the foundation of the central dogma, which is very important, but I don't want you to stress too much about that right now. I do want you to know that DNA codes for RNA and not directly protein, and I do want you to understand that RNA is going to code for protein. But other than that, that is our macromolecule PowerPoint review. Hopefully you're feeling a little bit better about your exam. You're working through that study guide, because remember, that study guide is your best tool in preparation. And if you have any questions, please ask. Thanks, you guys. Good luck.